comment on this, but racial disparities is not only a hot topic, but is a very important issue. And what we're finding is that in the context of different racial groups, the genetic predispositions don't look the same. And if you actually look at, for example, African American patients, there's a much higher frequency of uncharacterized variants, quote unquote, of the BRCA gene. And there's questions as to whether, what we typically think of as the mutations that are associated with risk in a Caucasian population are really very different in African American population. I've had the pleasure of working with some geneticists at Yale and who have been basically sequencing populations, founder populations, and what's clear is that the haplotypes in other racial groups are very different. And in fact, the genetic predispositions may need to be completely rethought in the sorts of testing that's done. I don't know, Nadine, if you want to say anything further.
So Lyndsay, I'm going to ask you to do something because it's really useful for the audience. Keep the microphone for a second. So this is Lyndsay Harris from Yale. So your last sentence in English for the audience. Sequencing, haplotypes. Why don't you tell everybody what we're talking about here?
Oh,
I don't mean to put you on the spot, but
that's why I was deferring to Nadine. So the composition of the chromosomes.
There you go.
And the genes.
So let's stop for a second. The audience, you may not know this, but you can now walk down the chromosome essentially. Imagine the chromosome. I always use 2nd Avenue in Manhattan. That's my base, right? So imagine one chromosome is 2nd Avenue from 14th Street to 96th Street. You can walk down each inch of that now, and you can actually look at what's at each spot. This was a tour de force. You couldn't do this 30 years ago. You'd have a postdoc 20 years ago spend how long doing this? I think you gave an example at the meeting we're at, David. What would it take? A lifetime, right? And now you can do it in? An hour. Okay? That's the convergence of informatics and computing technology and others. So that's what we mean by sequencing, right? Good. You want to finish? No, I just go ahead.
Right, so...
This is the only way I understand this, by making it really simple.
No, you're absolutely right, and I thank you for pointing that out. So the actual sequence of the genes vary by race. And if you look at the sequence of genes from an African population or a Hispanic population, even though they're still considered normal variants, they are very different from one race to the other. And so the predisposition for particular genetic mutations that predispose to breast cancer, for example, may be very different depending on the racial group. So if you're looking in an African American population for a BRCA mutation, you don't see it, it doesn't mean that it's not there. It just means that we may not be looking at it in the correct fashion.
Great. Right. So while you're passing the mic down to David, who I think had a comment, I'll point out that among the many BCRF-funded investigators is Fumi Alapati, who is doing exactly this kind of work in Africa, a very underserved area, and has identified a very high risk of the kind of breast cancer we see typically with BRCA mutations in North America, and is exploring that population to understand why they look different. It isn't only that they're underserved. It isn't only that they're not living in a Western diet. They actually look different, and understanding how that's associated with race or other aspects of ethnicity. Race is just one aspect, is, I think, an exciting opportunity for us to then intervene and make a difference. David, sorry.
Yeah, my name's Livingston, first name David. I work at Dana-Farber Cancer Institute, and I wouldn't be here without the extraordinary generosity and support and confidence given to us by Evelyn Lauder and this beautiful foundation, which is unique in my opinion, because they make it easy to do really hard things in a laboratory and in a clinic that the United States government won't do. Hats off to you.
Thank you.
So, I just want to make one comment about BRCA1 testing to our kind friend in the corner. Sometimes the answer is yes, there's a mutation. Sometimes the answer is no, there's not a mutation. The outcomes of those two decisions are a little clearer than what's usually the case when there might be something wrong, which is maybe there's something wrong. And the maybes outnumber the yeses by a lot in the country. Many women whose families include a history of breast cancer and or ovarian cancer going back a generation or sometimes two will have such a test, and the majority of them do not have proven BRCA1 mutations, but the test comes back maybe. So what we're working on, Mrs. Lauder and our colleagues and a bunch of us at Dana-Farber, is trying to understand what the maybes mean. Because it's clear that with the right ability to convert that maybe into a yes or no, one can, improve the care of those for whom maybe is yes, and relieve a heck of a lot of anxiety for those individuals for whom the answer was maybe, but it's really no. And at the root of the test is the fact that the BRCA1 gene, when it's really yes, not maybe, and certainly not no, has two outcomes. It means disease only in women. Men inherit mutations in the BRCA1 gene. They get off scot-free. They do not become ill. Only the women. So why only the women will actually help us figure out why the maybes exist and whether they mean yes or no. And the second one is, it's only the women and it's only two diseases, breast cancer and ovarian cancer. And why no other tissue of the body is affected is also a great mystery. And by trying to understand the maybes, we're actually beginning to gain a little insight into why the breast, and not the cervix, and not the uterus, and not the skin. And only these folks, only the BCRF make those kinds of experiments possible. I thank you.
Thank you, David.